Alright, so in this video we're going to talk about protection of the brain. If you could kind of feel the consistency of brain tissue, it would feel like a ripe avocado. That's how soft your brain tissue is. So there are three major factors that protect your brain. For one, we have your cranial bones. Two, we have the meninges. And the meninges cover and protect the central nervous system, so they surround your brain and spinal cord. They also protect blood vessels and enclose these venous sinuses, which we'll describe here soon. The meninges also contain cerebrospinal fluid, which is the third protective function here, because cerebrospinal fluid forms a nice fluid cushion for your brain to kind of bounce around within. Now, meninges also partition the brain within the skull. And the meninges are made of three major layers here. We have dura, arachnoid, and pia mater. So if you look at cross-section of the skull here, we'd find the skin of the scalp, some of the subcutaneous tissue, and then right here we have like the bone of one of your cranial bones, like let's just say the parietal bone. So you can tell it's a cranial bone because it's spongy bone that's found between two plates of compact bone here. Now just deep to the bone, what we'd find then are meninges here, because this is now within the cranial cavity. So you can see your brain right here with your cortex, that's gray matter, and then the white matter that's deeper. And then basically the meninges would help to affix the brain to the inner surface of the skull itself. Now the meninges are made of three major layers here. We have dura mater, arachnoid mater, and pia mater. And you can follow the dura mater out. Like you see that this layer of dura, it's called the periosteal layer of dura, would surround the whole inner surface of the skull. Deep to that, we actually would find this other layer of dura, which is called the meningeal layer of dura. So this is the meningeal layer, and you can see that it's a little deeper, but it also can separate from the periosteal layer in these kind of special areas here, where you'd find that right here, there's a separation of periosteal layer of dura mater and the meningeal layer of dura mater, and in that place where they separate, there's now a blood-filled space. Now this blood-filled space is called the superior sagittal sinus, and it's an example of one of the venous sinuses that you find around in the meninges of your brain. Ultimately, the function of these venous sinuses is to allow for some drainage of blood from your brain back into these spaces that eventually go back to your heart. But it also serves as the reabsorption site for cerebrospinal fluid. So you can see this little protrusion up here into the venous sinus. This is something called an arachnoid villus, or arachnoid granulation. And essentially what this is a little patch of arachnoid mater that is protruded up into the venous sinus, and this is where cerebrospinal fluid gets reabsorbed. So if you look at this whitish layer here, this is the arachnoid mater. And it's called arachnoid mater because it looks like a spider, kind of spiderweb-like appearance here. Now the space here between the pia mater, right here, and the arachnoid mater, this space is called the subarachnoid space. Now there's a space here because it's actually filled with cerebrospinal fluid. And that cerebrospinal fluid helps to surround the outer surface of your brain between the arachnoid and pia layers of the meninges. And that cerebrospinal fluid helps to cushion the brain, but within the meninges. But it also helps to nourish the brain because CSF is made of a lot of like water, sugar, and electrolytes. Now it's constantly being formed, and it needs to be reabsorbed. So the places that CSF gets reabsorbed back into are these arachnoid granulations, or arachnoid villi, which are essentially protrusions of the arachnoid mater up into these dural venous sinuses. So cerebrospinal fluid gets reabsorbed back into blood. So it's made from blood and it gets reabsorbed back into blood. So just to summarize those three major layers, remember we have the dura mater, arachnoid mater, and pia mater. And the pia mater means soft. And mater means mother. So soft mother is the pia mater, mostly made of an outer layer of blood vessels and areolar connective tissue that essentially help to surround the outside of the brain and nourish the cortex from the outside. Now there are some spaces here that are important to note as well. So there's a space right here between the meningeal layer of dura and the arachnoid mater called the subdural space. Now the subdural space is a potential space because it normally doesn't exist unless it's filled with something. So blood can fill this space, tumors can fill this space, even cerebrospinal fluid can. And so the subdural space is a potential space that can potentially get filled with a fluid like blood. So if you've ever heard of a subdural hematoma, it's basically a leakage of blood into this space right here. Remember, we have the subarachnoid space, which is not a potential space, rather it's usually always filled with cerebrospinal fluid that serves as a nice protective cushion. Now what's interesting too is if you look closely, you can see here that the meningeal layer of dura can come together and essentially dive down deep between the hemispheres of the brain here. Now, these protrusions here, this example here is called falx cerebri. And this is an example of areas of dura that help to separate some of the hemispheres of your brain. Now, the dura mater is the strongest of the meninges, and it's made up of two layers of fibrous connective tissue. Remember, we have the periosteal and meningeal layers. Periosteal layer is found attached to the inner surface of the skull, but it's only found around the brain. The meningeal layer is a true external covering of the brain. And this is what extends also into the vertebral column to surround the spinal dura as well. Now, the meningeal layer, when it separates from the periosteal layer, can form those dural venous sinuses, which are basically the spaces that collect venous blood from your brain, and they empty back into the jugular veins that drain that blood into your neck. Now, the dura mater extends inward in several areas to form these flat partitions called dural septa, septa meaning like separation point. And there's three main septa around the brain. We have the falx cerebri, falx cerebelli, and tentorium cerebelli. So what this shows is an example of falx cerebri, and it's essentially the dural septa that would separate the two cerebral hemispheres, right versus left. We also have the tentorium cerebelli, which would separate the cerebellum from the cerebrum, and the falx cerebelli that would separate the two cerebellar hemispheres. And essentially what these are made of are extensions of the meningeal layer of dura that dive down deep. And you can find here that these blue spaces are the dural venous sinuses. So there's an example of dural venous sinuses, throughout the dural septa. So you find the superior sagittal sinus here. We have the inferior sagittal sinus. We have the confluence of the sinuses here, which eventually drain back into the jugular veins that exit the skull and then drain that blood back towards your heart. Now, this is showing in a cadaver dissection, what the meninges would look like. So you can see your cranial bone here is pretty thick, and that's a nice protective layer. We also have some of the dura mater here. So you can see the periosteal layer of dura. You can see the dural venous sinuses as well from a posterior view. So here's superior sagittal sinus meeting up with transverse sinus to form the confluence of the sinuses here. And in this type of dissection, what they've done is they've actually filled the blood vessels with a plastic type of rubbery material that makes these look more distinct. Now, in a living specimen, they wouldn't look this blue. These have been injected with a dye. So the arachnoid mater, if you remember, is deep to the dura mater. It's the middle layer with a spider web-like extension. And they're separated from the dura by the subdural space. Subdural space, remember, is the potential space that could fill with blood if you have a hematoma. But the subarachnoid space is located just deep to the arachnoid mater. It contains cerebrospinal fluid and essentially helps to cushion the outside of the brain by forming that nice protective space there. Now, there are areas where arachnoid mater protrudes into the dural venous sinuses, forming what we call the arachnoid granulations. And this is the place where CSF gets reabsorbed back into the venous blood. Pia mater, if you remember, is a delicate connective tissue that clings tightly to your brain, and it's following every convolution of the outer surface of your brain. It contains lots of tiny blood vessels that effectively feed this outside of your brain. So I think of the pia mater as like an outer nourishing blanket that surrounds the cortex of your brain. So just to summarize this again, remember we have our dura mater, arachnoid mater, pia mater. Dura mater's got two layers. We have periosteal layer and meningeal layer. And then what separates the dura from the arachnoid mater is the subdural space, which is only a potential space, so it's not normally there unless it's filled with something. Then we have our arachnoid mater, which has just deep to that has the subarachnoid space filled with cerebrospinal fluid. And then we have our pia mater that clings tightly to the outside of the brain. Now, cerebrospinal fluid forms a liquid cushion that maintains a fairly constant volume around the brain. Now, CSF doesn't only just protect the outside of the brain, because you also find the CSF filling the inner surface, the inner chambers of your brain, which are the ventricles. So what's cool about CSF is it actually makes your brain buoyant within the skull. So it reduces the weight of the brain by 97%. So effectively, the brain floats almost entirely within the skull, and so there's almost no weight on the base of the brain because it's floating within your cranial cavity. Now, the function of CSF is to protect your cranial central nervous system from blows and other trauma, but it also nourishes the brain because CSF is a lot like Gatorade because it's made of water, sugar, and electrolytes. It's formed from blood plasma, but it has a lot less protein. It also has different ionic concentrations than plasma. Now, all CSF is made by your choroid plexus, And choroid plexus is these clusters of capillaries that hang from the roof of each ventricle. They're enclosed by pia mater, and they're surrounded by ependymal cells. Remember, the ependymal cells were a glial cell that made CSF, and these ependymal cells basically use ion pumps to control the composition of CSF and help cleanse CSF by removing wastes. The cilia that are on these ependymal cells help to wave that CSF around and help to circulate that through the ventricles and around your brain. Now, normal adult CSF is only about 150 mils. So that's, there's not a whole lot of this stuff surrounding your brain, but it's replaced every 8 hours, which keeps it pretty fresh. So if you look at the direction of CSF flow within and around the brain, you'd find that it's made in all the ventricles of your brain. So you find choroid plexus in the lateral ventricle, you find choroid plexus in the third ventricle, and that's what's shown as red here. This is where CSF is being made. So CSF would flow from the lateral ventricle into the third by the interventricular foramen, and then from the third ventricle it could flow into the fourth through the cerebral aqueduct. And then the fourth ventricle, CSF has two ways to go here. It can continue down the spinal cord through central canal, or it can exit the brain through an aperture here, and then surround the outside of the brain by entering the subarachnoid space. So all of this kind of light blue here shows the CSF that surrounds the outside of the brain in subarachnoid space. So you're between the arachnoid and pia mater here. And remember that CSF must be reabsorbed back into blood. So you can see that this shows the arachnoid granulations where CSF is reabsorbed back into the dural venous sinus, and then from the dural venous sinuses they all come back together and then drain towards the jugular veins. Now this is showing an example of choroid plexus where you find a blood vessel here with our red blood cells. There's some pia mater that surrounds these blood vessels, but they're lined with an epithelial type of lining which is actually made of glial cells called ependymal cells. So what these ependymal cells do is they form CSF by creating something called a filtrate that contains a lot of glucose, oxygen, vitamins, and ions. And I think of this as being very similar to Gatorade because it's a lot of water, sugar, and electrolytes. And so it's actually modified blood plasma. Now this would actually fill the ventricles of your brain and then basically flow throughout the ventricles because they're waved by the cilia here that are on these ependymal cells. Now, choroid plexus also helps to reabsorb wastes and other unnecessary solutes, which keeps your cerebrospinal fluid clean. Now, what's interesting too is we find something called the blood-brain barrier throughout all the major blood vessels in your brain. Now, the function of the blood-brain barrier is it helps maintain the stable environment for the brain, because there are things in your blood that could potentially be harmful to your brain tissue, and this can lead to uncontrollable neuron firing and possibly even neuron cell death. So the blood-brain barrier is exceptionally impermeable to a variety of different substances. And this blood-brain barrier is made by three major factors here. First of all, we have a continuous endothelium of our capillaries. So the blood vessels that traverse the inside of your brain are very tightly interlinked, their cells are, through a lot of tight junctions. We have a thick basal lamina that surrounds these capillaries and it forms a nice protective sheath. And then there's also the foot processes of astrocytes that surround the blood vessels and they help form an additional barrier here. So if you look at the blood-brain barrier, effectively what we see here is for our blood vessel, you'd find our endothelium cells that line the inside of this blood vessel, and those are very tightly bound together by tight junctions. We would find the underlying basement membrane that forms a nice protective outer layer that also traps debris and prevents that from getting the brain. And then those blood vessels are also surrounded by the foot processes of astrocytes, which are the third layer here, third line of defense, that help to ensure that chemicals in your blood don't just leak out into your brain tissue. So the blood-brain barrier is selective, but it's not absolute. So what's interesting here is the blood-brain barrier can allow certain nutrients to move by facilitated diffusion, but other things like metabolic wastes, proteins, toxins, and most drugs, and small and non-essential amino acids and potassium, they're all denied. These are all things that could potentially harm your brain, so the blood-brain barrier keeps those out. But any fat-soluble substances like alcohol, nicotine, anesthetics, and also small molecules can typically cross the blood-brain barrier. Now, there are some parts of your brain that have a very thin or absent blood-brain barrier, and these are important because you need areas of your brain to monitor the chemical environment that's in your blood. And so you find this in places like the vomiting center of your hypothalamus. That way, if you have toxins in your blood, you can initiate a vomiting reflex to help to, potentially clear out those toxins from your digestive tract.